Hello and welcome to my today's presentation. I'm Lisa Mais and I'm a PhD student in Dagmar Keimüller's lab at the MDC in Berlin. Today I'm talking about PatchPerPixMatch, a method for automated 3D search of neuronal morphologies and light microscopy data. The work is based on two large projects done at Genelia, Howard Youth Medical Institute, and aims at bringing them together. So the first one is the FlyEM project, where they released the hemibrain connectome. Here they imaged one fruit fly brain with electron microscopy and traced all neurons and synapses in it. On the other hand, there's the FlyLight project, which aims at providing a large resource of transgenic lines for targeted neuron manipulation and imaging with light microscopy data. On the left side here you can see the full expression pattern of such a line, and the two right images are multicolor flip-out acquisitions, which is a stochastic labeling technique to only show a random subset of the full expression pattern. And to have a good coverage, actually each line is imaged multiple times. And now we want to combine the information which we can get from both modalities, which is the connectivity from the EM data and the neuronal function from the LM data. Another goal is the creation of so-called split GAL4 lines. In more detail, if we find two different lines which have one or more neurons in common, then this information can be used to create a new, much sparser transgenic line, only showing the intersection of the two original ones. Then I want to jump into the neuron matching part, and here the scenario is as follows. We have a given target neuron identified in the EM data, and we want to look for this neuron in all multicolor flip-out samples, or short MCFO samples. And to this end, we developed a method which we called PatchPerPixMatch, which mainly consists of two steps. The first one is the segmentation of the MCFO light microscopy data, and the second step is the actual matching. Before coming to the segmentation, I want to give you a closer look to the data. Here you can see that the data is in 3D and multicolor, and also the number of neurons varies a lot, so this can be any number between 1 and 50. The data is quite challenging as the neurons are very thin and have complicated shapes, and also they may overlap or run in parallel. And another fact is, which we need to keep in mind, that the manual annotation for this data is only possible for sparse volumes. Considering these challenges, together with Peter Hirsch and Dagmar Keimüller, we developed PatchPerPix, a deep learning-based method for instant segmentation that can handle arbitrary shapes and overlaps. And at the time of publication, this method achieved state-of-the-art results on 4 benchmark datasets, which are cell segmentation in 3D and 2D light microscopy data, then the elegant warm bodies in the dish crawling above each other, and the ISB 2012 EM challenge. Then we applied PatchPerPix to the FlyLight data, and here you can see some qualitative results of it. This is a relatively easy example with only a few neurons. Here you can see the raw data, the manually annotated ground truth data, and in comparison to that, the prediction obtained with PatchPerPix, where we actually obtain quite nice reconstructions of the neuron. And here's a second example with slightly more neurons. Then I want to continue with the matching. We want to check if the given target neuron does occur in a certain MCFO sample. So in the first step, we got our fragments, and we yield large fragments, but we cannot consider them as complete reconstructions. That's why we need to handle false splits and false merges in the second part of the pipeline.
For this, we formulate the matching as a global optimization problem where we want to find the best fragments to cover the given target neuron. We take into account the similarity in terms of n+ score, then the coverage and the color difference. By doing this, each fragment gets assigned the similarity and the coverage, and the color difference is used as pairwise term, and then we want to find the fragment selection that maximizes our energy. And at the end, each MCFO sample is assigned such a score, which says how well the fragments can actually cover the given target, and then we can sort all the MCFO samples according to the score and can provide the best matching samples as matching candidates to the biologists. And here you see the cutout of the raw data at the position where the selected fragments are, and here you see it together overlaid with the target neuron. We were running the PatchPerPixMatch pipeline in total 1.8 billion times. For the brain, we had 30,000 target skeletons from the hemibrain connectome, and were looking for them in 40,000 MCFO samples of approximately 3,500 transgenic lines. And we did the same for the ventral nerve cord, where we had 23,000 target skeletons, looking for them in 26,000 MCFO samples. Then I'm coming to the evaluation. Here the problem is that we can only evaluate already known matches or depend on an expert to determine if a match looks promising or not. In the following figure, Alyosha Neuron annotated true hits within the top 100 search results for 10 different cell types. And in more detail, he assessed the line rank, which is the best matching MCFO sample per line. He did this analysis for PatchPerPixMatch as well as for color depth map search, which is an alternative approach developed by Hideo Otsuna at Genelia. So in the figure, you can see the search result rank in the x-axis, which goes from 1 to 100, and the corresponding number of matches in y. And in the first plot, you can see the curves inserted for all 10 cell types plus the average in bold. So PatchPerPixMatch, which is depicted in magenta, performs slightly better than CDM in this set of target neurons, but it strongly depends on the cell type, as you can see in the two other plots. So for the cell type LC18, PatchPerPixMatch finds significantly more matches than CDM within the best 100 matching samples, but for CT1 it is exactly the other way around. So you can really consider both methods as complementary, as the matching candidates are quite different. And finally, we made our search results available in two different ways. We created PDFs showing the top 150 matches for each target neuron, which is available at pppm.janelia.org. And our search results are also integrated into the web application Neuron Bridge, which is available at neuronbridge.janelia.org. And here the user can actually choose between the results from PatchPerPixMatch or color depth map search. At the end, I want to say some words to our future work. As we have seen, PatchPerPixMatch works quite nicely, and we have generated a lot of data where biologists can browse through it and find matching candidates, but still we want to further improve our method. And we can do this by either tackling the segmentation or the matching part, and as we have manually designed components in the matching, we want to start there. In more detail, we want to use graph neural networks, where we construct a fully connected graph with the nodes representing the PatchPerPix fragments. And then we can use the NBLAST and coverage and color as node features, and as edge features we want to use the overlap, which means that different fragments may cover the same EM point, and this is represented in the overlap. And although we are in a very early stage of the project, we already get quite promising results. I want to close my presentation with acknowledgements. I want to say thank you to all the people in the Keinmüller lab, especially my supervisor Dagmar Keinmüller and my colleague Peter Hirsch, and all the collaborators at the Genelia Research Campus, especially the Flylight project team with Geoffrey Meissner, the project technical resources and scientific computing. Thank you.